In the last worksheet, the last video, we looked at a relatively straightforward problem in population genetics. I gave a little review of what population genetics is and how we get these particular numbers or these equations, or the Hardy-Weinberg equations, and the principles of how we use them. So if you missed that, go back and watch the previous video, so that we're going to just pick up from there. Because now here's another worksheet. So what this one is here we're considering a flower whose petal color is determined by the locus G. Two codominant alleles, big G and little g. So remember what codominance means is that our homozygotes, dominants, our heterozygotes, and our homozygote recessives all have their own phenotypes, that the heterozygote is going to be some intermediate phenotype to the two homozygotes. Okay, so we can see that in that big G, homozygote dominant, confers red petals. Big G, little g, or our heterozygotes, gives us pink petals. Little g gives us white petals. Okay, so you count all the so now what this allows us to do is we can look in a population of these flowers and we can actually count how many homozygote dominants we have, how many heterozygotes we have. We can distinguish the two. And so we count up and we have 70 red flowers, 20 pink flowers, and 10 white flowers. And then, so if the population is assumed to be in Hardy-Weinberg equilibrium for this locus, what are the allelic genotypic frequencies? Okay, so the first thing we're going to do is just say, okay, let's just assume this is in Hardy-Weinberg equilibrium. Let's calculate what we would expect our genotypic frequencies to be. And so remember, what we're going to be doing for that is we're going to calculate our allelic frequencies. From those allelic frequencies, we can put them into Hardy-Weinberg's equation with p squared, 2pq, and pq squared to get our predicted or expected genotypic frequencies. And then the last question is what it's going to say is this population actually in Hardy-Weinberg equilibrium? And at which point what we'll do is we'll calculate the actual genotypic frequencies of this population, and we'll test to see whether or not our expectations are meeting our observations. And if they are, then yes, it is. And if they're not, then no, it's not. Okay, so to calculate the allelic frequency of the big G, what we're going to do is we're going to look at the part over the whole. And so when we're looking at the part over the whole, we want to know how many of all of the alleles, how many are big G, or what proportion of them are big G. Now, what we need to add up here is, so we have 70 red, 20 pink, and 10 white. So like the last problem, we have 100 individuals. Makes our math easy. Because each individual has two alleles, we have 200 alleles. Okay. Now, the frequency of the G allele. So what we want to know is the frequency of big G, which is also equal to P, is going to be the number of big G's we have divided by 200. So the number of big G's we have is, here we have 70 red. So remember, red petal flowers have two big G's. They're homozygote dominant. So we have 2 times 70. And then big G, little g gives us pink flowers. So the pink flowers that we have a single big G. So we want to add 20 to that, which would give us 140. 140 plus 20 is going to give us 160 over 200, which will give us a p-value of 0.8. Okay. So now, to calculate the little g allelic frequency, what we're going to do is we're going to say the frequency of little g is equal to q. P plus q is equal to 1. So if we have 0.8 plus q is equal to 1, subtract 0.8, subtract 0.8, and we will get q is equal to 0.2. So the predicted frequency of our homozygote dominance, well, that's the frequency of big G, which is equal to p squared. So if we take 0.8 and square it, we get 0.64. Okay. Same thing if we, for our predicted heterozygotes, well, the frequency of big G, little g is equal to 2pq. So that would be 2 times 0.8 times 0.2. And so if we take 0.8 times 0.2, that's 0.16. 0.16 times 2 is going to be equal to 0.32. Okay. And for the last one, it's more of the same, in which what we're doing is we're going to calculate the frequency of little g, which is equal to q squared. And so then we're going to say, well, q squared is 0.2 squared. So if you square 0.2, you're going to get 0.04. And so these, it's important to note, are our predicted frequencies. And so, as the problem explains, or then the next part of the problem asks, using this information above, determine whether or not this population is in Hardy-Weinberg equilibrium. So remember, we have 70, 20, and 10. We'll move that information down here so that we can see it. So we have 70 red, which are big G. We got 20 pink, which is big G, little g. And we have 10 white, which is little g. And so to calculate this frequency, all that we have to do of our actual, this is our observed genotypic frequencies. We just divide that number by 100 because we have 100 individuals. So 70 divided by 100 is equal to 0.7. This would be 20 divided by 100, which would be equals 0.2. And this is 10 divided by 100, which is 0.1. And then we compare. And we say, does this equal that? No, it doesn't. Does this equal that? No, it doesn't. Does that equal that? No, it doesn't. So, no. Not in Hardy-Weinberg equilibrium. Okay, so that's how we test. You take the actual number of individuals that you have, calculate your allelic frequencies. From those allelic frequencies, you calculate what your predicted frequencies would be. Once you've calculated your predicted frequencies of genotypic frequencies, you calculate your actual genotypic frequencies. Then you compare your predicted or expected genotypic frequencies with your actual or observed phenotypic frequencies. If they match, it's in Hardy-Weinberg equilibrium. If they don't match, it's not in Hardy-Weinberg equilibrium. Now, in real life, what you would have to do to this is you would apply some sort of statistic, like a chi-square test, to demonstrate that you do not have, or that there isn't statistically that this is not, your expectations are not meeting your observations. But for this purpose, we're not going to do that. And so that's how you would solve that problem of actually testing a population to see whether or not you are in Hardy-Weinberg equilibrium. So I hope this helped. Let me know. Thanks. Have a good day.